Good evening and salud to everyone imbibing. I hope you're enjoying the Pisco Sours. I've heard they're strong. I'm Michael Dubray, manager of adult programs here at the Dallas Museum of Art. I'm delighted to welcome you all to today's 𝗕𝗊𝗆 Family Lecture Series. The lecture series is generously sponsored by the 𝗕𝗊𝗆 Family Foundation for the purpose of presenting programs designed to stimulate interest in and advance the scholarship in the art and archaeology of the ancient world. Although our dear friends Ned and Raynette Bichelle and Betsy Todd could not be with us today, we want to thank them for their continued support of this program and our work at the Dallas Museum of Art. Before proceeding, I'd like to recognize the Honorable Freddy Martinez, Deputy Consul General of Peru. We are honored to welcome you and your staff to the museum. Additionally, I'd like to give a very special thanks to 𝗕𝗵𝗵𝗵𝗆 for the wonderful traditional Peruvian guitar music in the gallery. Today's program highlights the groundbreaking research and technical analysis of one of the most significant extant painted Peruvian textiles and the first exhibition of its kind. We're delighted to have Cory Dodson and Cory Rogge, conservators from the Menil Collection, share their research conducted in conjunction with the DMA's curator, Dr. Michelle Rich, and objects conservator, Fran Bass. Our moderator this afternoon is Dr. Michelle Rich, the Ellen and Harry S. Parker III Associate Curator of Indigenous American Art. Since joining the DMA in 2018, she has worked to highlight the depth and diversity of Indigenous art through the Americas through exhibitions, publications, and gallery installations. Her curatorial practice bridges archaeology, art history, and museum work with a focus on rigorous scholarship and amplifying Indigenous voices. She edited the Arts of the Ancient Americas at the Dallas Museum of Art, a 360-page catalog featuring new research and over 100 new photographs of highlights from the DMA's Indigenous American Art Collection. She's currently developing an exhibition about Maya women that features both ancient and contemporary art. Dr. Rich holds a PhD in archaeology from Southern Methodist University and has been affiliated with the U.S. Guatemalan Proyecto Arqueológico 𝗪𝗺𝗺𝒻 since 2003. If you've not visited the exhibition, make sure to do so as it ends tomorrow. Please join me in welcoming Dr. Michelle Rich to the stage.
Good afternoon, everybody. Can you hear me? Excellent. It's so good to see so many folks here on this beautiful Saturday afternoon. Thank you, Michael, for the lovely introduction. And I'm just really excited to share this information with you all today. Fragmented Mysteries, the history of the Prisoner Textile, gives us an opportunity to dive deep into one of the most important pieces of painted textile that we are currently aware of from the Chimú culture of Peru. We have been very fortunate at the Dallas Museum of Art to partner with our colleagues at the Menil Collection, Dr. Cory Rogge and Cory Dodson, on this project. It began in 2021 when Fran Bass and I visited the Menil to see their exhibition called 𝗏𝗏𝗏𝗏, Visual Histories of the Central Andes, which included the Menil's section of the Prisoner Textile. So fast forward to this photo of Kari, Fran, and myself taken in October of 2023 as Kari conducted experimental eraser testing on the DMA's fragment of the Prisoner Textile. They will dive into that when they're up here on the stage, but I just really personally love this photo because it demonstrates some kind of hive mind that curators and conservators share, like chunky glasses and black. And then, of course, when we're all up here on the stage together, please note. So my job here today is really to set the stage for our guest speakers by providing background and history. So let's get into it. Any excuse at all to show this picture of Nora Wise with Sir Lancelot the Llama. That's a double L, Lancelot. So for the DMA, this story actually starts with Nora and John Wise, and the acquisition of the 𝗧𝗍𝗃 collection. Around 2,000 objects of various materials were acquired from the 𝗧𝗍𝗃s, including the Prisoner Textile, between 1976 and the mid-1980s. All of the DMA works in Creatures and Captives were part of the 𝗧𝗍𝗃 collection. This acquisition was transformational for the museum, and upon receiving portions of the gift, they immediately went on view, and we can see here in 1977 and 1980 the different ways in which the materials were exhibited. And you can see in the circle on the far left, that is our section of the Prisoner Textile. There's another painted textile that's laid out on the floor. And then we also have a gold spangled tunic, which you can see in the Indigenous American art galleries on the 4th floor if you want to go have a look in person. There are over 650 textiles in the 𝗧𝗍𝗃 collection. They are woven, they are embroidered, they are painted. They comprise earrings, hats, tunics, shrouds. You can see that there's kind of a multi-material approach to creating textiles that ancient Andeans implemented. We've got these great earrings. I wish we could replicate them for the gift shop. In the lower left-hand corner with silver and the long fringe and feathers at the tips. How awesome would those be? It is a little bit daunting to work with this quantity of textiles, especially when I'm a Maya archaeologist and I work in subtropical environments and textiles do not preserve. So this has been a learning curve for me as a curator, and it's been a journey that I have been supported on by knowledgeable colleagues and associates at museums and archaeological projects. Ancient textiles were an important technological development in the ancient world. Clothing of many kinds was developed, but of course, that extends out into tools, such as nets for fishing, slings for hunting and warfare. Wari and Inca cultures in the Andes used textile quipu to store information. So textiles were they were physically important, they were also logistically important, and they were also symbolically important. In the Andean region, textiles are some of the most prized objects, valued even more highly sometimes than silver or gold. So they had a different value system that they attached to works of art and objects in their cultural milieu than we have today. And so I think we have to keep that in mind. Because of the aridity of the Peruvian coastal deserts, these organics did not disintegrate, which is why we have such an amazing opportunity to study the textiles from that region. Ancient Andean weavers created their textiles primarily with cotton and camelid wool. And again, here we can harken back to Sir Lancelot, the llama, and thinking about the camelid wool, which would have come from vicuña, alpaca, guanaco, and llama. So they had this range of animals that would provide them with this wool. Are there any weavers in the crowd? Please raise your hand. Don't be shy, weavers. I see a weaver. I see 2 weavers, yes. Weaving is fascinating to me. I am learning about this as I go, and spinning yarn is like a mystical black magic. I don't know how people develop this technology, but spinning with a drop spindle requires one to be able to multitask with both hands. It takes training and practice to develop coordination. There's concentration is important. If the yarn that you're spinning is too thin, the fibers won't have enough to hold onto and the yarn will break. If it's too thick, there won't be enough tension and the yarn might unravel. There's a whole host of things that can happen while you're spinning threads. And so I just want us to recognize the like amazing capacity of human ingenuity that we see when we're looking at these textiles. How do we get finished textiles? There are multiple techniques. We've got fusing, so think like felted things, like your dryer balls that are in your dryer. There's looped approaches, so think crocheting and knitting. And then there's woven, which is technically anything that's made using a loom. Ancient Andean people, and also across the Americas, used the backstrap loom. So with the backstrap loom, you wear a strap around your waist and you attach the far end to something stable like a tree trunk or a pole that allows you to keep the kind of tension that you need to weave. And this is a practice that is still alive and well today. It's not disappeared, and it's in fact a very vibrant industry in the Americas. I'm just giving us all like a 101 in textiles so we can all be on the same page when Kari and Cory get up here and wow us with their science. So the terms used to distinguish threads that go vertically versus those that go horizontally in a woven textile are warps and wefts. And a friend of mine, Herman Cardona, he taught me this trick to remember what a warp is versus a weft. So warp strings go vertically like the strings on a harp, and weft threads go left to right, so warp, harp, weft, left. Couldn't remember it otherwise. So, how do we get from woven designs to painted designs? Let us look at these two close-ups here of textiles that are in the collection at the DMA. On the left, you see a very close-up of some iconography on a very beautiful tunic, which is small and in the lower left-hand corner there. The yarn was dyed first, and then the design was probably plotted out, but created as it was woven. And these woven designs are pretty standardly geometric. You don't have a lot of flexibility to create curves or any kind of more fluid design when you are weaving. So you're very much restricted by that. And on the right, we have a plain undyed cotton cloth that was woven first and then dye was painted on. And this is a piece that's upstairs in the textile gallery in Creatures and Captives. We have 3 figures here. You can only see 2 in this detail. And you can see how expressive and rounded and fluid and free the painted textiles are in comparison to the woven textiles. We do get repetitive design motifs and iconography in both woven and painted textiles, birds, fish, humans, mythical beings, step frets, and other geometric patterning that appear. Think about living in a coastal region and you would be influenced by the flora and fauna around you as you created these designs. The figures in the painted textile, we thought about maybe having our social team post that for Halloween because they're kind of scary and clown-like, but we decided not to. We have no idea what the textile means. Take a look at it if you go upstairs after the talk and just come to some conclusions for yourself. That's one of the interesting things about these works. You can really just spend a lot of time close looking and come away with just some fascinating thoughts about what the textiles might have meant or how they might have functioned. So let's think about some insights here that we've garnered in my very short presentation. Ancient Andean artists have been painting textiles for centuries, and they have not been very well studied. I'm going to say this because historical inertia in academia and scholarly research has really favored the dyed woven textiles. The colors are more vibrant, there's more examples of them available in general, and they have really taken center stage. So this is a really fabulous opportunity to collaborate with the Menil to dive deep into our painted textile collection. These, I don't know if red-headed stepchild is still a phrase that you are supposed to use, but that's really what these textiles have been, and that's why Kari and Cory's research is so amazing. We also have done some other work on our painted textiles in the collection here, and this is a photo of Fran Boss using XRF on one of the textiles that's also on view upstairs. It had metal fragments stuck to it, and so Fran was able to identify the elements creating the metal. It was silver, copper, and iron, right? Yes. And what that suggests to us is that this textile was in an archaeological context with metal objects. And so if the metal was corroding, because copper corrodes, and the textile was touching the metal, then we would get some transference of the metal to the textile. So this, looking at the fine clues that we see in the textiles can really help us learn a little bit more about their life histories than we currently know, because many of them were not properly archaeologically excavated. I also am kind of thrilled. This is also another large textile that's on view upstairs, standing frontal figures and birds. In this whole textile, there is one bird holding a cutie little worm in his beak. And we had a great drawing of that was done by a previous curator here, Carol Robbins, and we were able to, Hayden Jaraska, research assistant for Indigenous American art, transcribed or redid that drawing, and so we were able to bring that into the gallery and kind of do like a hide and seek, seeing if you can find it. You all have a clue here, though, so you're cheating if you go upstairs and you're like, oh, look at the worm. No. So one of the things that I want to flag before I forget is that we produced a really awesome brochure that you can pick up outside on your way out of the lecture that talks about the whole process of creating creatures and captives and working with Kari and Cory and the team at the Menil. It has been a journey that started many years ago, and I think sometimes when folks come to the museum and they see our awesome exhibitions, there's not really an understanding of how long it takes to produce something, even a small exhibition like Creatures and Captives, especially when you're incorporating novel research into the exhibition process. So on the left is the Menil's fragment of the Prisoner Textile, which is a double panel, and on the right is the DMA's fragment. Right now, we have identified, not us specifically, but researchers who are interested in this textile have identified 10 fragments of this textile that are strewn across the world in museums, and together they comprise 13 different focal fields. So this is a very long textile. We'll get there in a minute, and I'll give you some examples of the length of it, but I just want you to, when I say fragment, I don't mean like a teeny thing. Our fragment is 6 feet by 6 feet. So we're talking big fragments. And this was a monumental painting done by Chimú artists that depicts prisoners of war and conquered peoples, we think. It was rediscovered around 1951 in a non-archaeological excavation. And we can see here John Wise looking very dapper. He acquired 2 sections of the textile, and one of them he gave to the museum. It was originally called something else, but now it's the Quai Branly in Paris, France. And honestly, I got a cold email the other day from a woman I do not know who follows the DMA on Instagram, and she wrote and said, I was John Wise's assistant, and I'm so excited that his collection went to a good home. I visited the Quai Branly in 1972 and saw the fragment that he gifted to them there on view at the time. I still have to answer her email, but amazing. So the world is like a smaller place than we think it is. This is the reconstruction that a colleague of ours, Andrew Hamilton, has proposed for the different sections of the Prisoner Textile. You start in the upper left-hand corner and move across the screen, and then go back down to the lower left-hand corner and move across the screen. And the lower left-hand corner shows our textile, and you can see the Menils is the second one on the top row. So this textile is truly monumental. Hayden Jaraska put together this comparative sample for you. So it's over 75 feet long, which is the size of an adult blue whale, or a tennis court, or an 18-wheeler, or 1/4 of an American football field. So this is pretty impressive in its size and the effort and labor that went into producing it. So think about that as we move into the next section of our talk. And I am more than thrilled to introduce Dr. Cory Rogge, who is the Director of Conservation at the Menil Collection and President of the American Institute for Conservation. She earned a BA in chemistry from Bryn Mawr and a PhD in chemistry from Yale, and held postdoctoral positions at the University of Wisconsin-Madison and the University of Texas Health Sciences Center in Houston. Before joining the Menil full-time in the summer of 2023, she served as the Andrew W. Mellon Research Scientist. Wait, did I get that right? Thank you. At the Museum of Fine Arts, Houston, and the Andrew W. Mellon Assistant Professor in Conservation Science in the Department of Art Conservation at the State University of New York, Buffalo State College. While much of her research focuses on 20th century artists, paints, and pigments, she works on materials across all cultures, media, and ages. And since 2014, Kari Dodson has been an objects conservator at the Menil Collection in Houston. I love this. She especially enjoys technical art historical research and using innovative imaging techniques to make visible the invisible. Superpower. It's awesome. She has held internships and fellowships at the Worcester Art Museum, Metropolitan Museum of Art, and the Barnes Foundation, and also the National Museums of Malawi. So before landing on a career in conservation, she taught ESL, worked on archaeological digs, and was employed at a nuclear reactor. We have some formidable women here today. Let us welcome them. Okay. We're going to adjourn to the central region here.
Hello, everybody. Welcome, and also thank you so much for having us here today. Michelle, that was fascinating. As I'm about to explain, we are always learning new things about this textile, and it's kind of amazing. I am sitting here today with Cory Rogge, who, as you learned, is the director of the Department of Conservation at the Menil. But more importantly for our talk this afternoon, she is also a conservation scientist, and that has been super valuable, as you will see. I think I also want to take this moment to acknowledge the contributions that the DMA have made, Dr. Rich, and also the incomparable Fran Boss, who is in the audience this evening.
Can you wave? Can you wave, Fran?
Their contribution, the close collaboration that we've had, the fact that we're only 4 hours away from one another has been really instrumental to making progress in our efforts. So, without further ado, let's talk about what we've learned about the Prisoner Textile in the last 4 years, and on a more general level, how it is that we go about a technical art historical study. I think I'm repeating a little bit of what Michelle said, but round about the time that the pandemic hit, there was a renewed interest in the textile. And the Menil's fragment was included in the 2021 exhibition that you're seeing on the screen here, curated by Paul Davis, our curator of collections. And this renewed interest really inspired us to apply for a Bank of America Art Conservation Project grant, which we were awarded. Yes, and their generous funding has allowed us to really lavish our piece and other fragments with, the latest in conservation and imaging and science technology. So it's been a collaboration with many, many people, as you'll see. Our most crowded slide is our acknowledgements slide at the end. You can wait for that. So we've worked with other institutions, but also individual scholars, and we're going to try to call out their contributions as we go through, because we appreciate what all of them have brought to the table. Let's get into it. Every big project like this, I feel like, starts with just a laundry list of broad questions for which we need answers. And the list gets longer as you go, but that's only natural. I thought we would use the list of questions you see here as kind of a roadmap for our time together today. And so we're going to talk through some of these questions and how we went about getting answers. Most of the examples I'm going to use are from the Menil Collections fragment, because that's the one I have the closest access to, but you're also going to see a lot of our work with the DMA. So buckle your seatbelts. Let's start with what is the fiber. So we began this whole project just looking really, really closely at the textile, and then adding in things like high-powered microscopes to let us see even more. The image that you're seeing on the screen is a close-up of a section of a little blue figure on the Menil's fragment. And not only can you see the individual yarns and the way that they are interlaced to create the textile, but you can also see a little bit, can you see the blue? I hope you can see the blue colorant on there. And you can kind of see how it's interacting with the fibers, and that's a really important clue to what we're looking at. Another shout out I have to make now, Kathleen Kiefer, who is a textile conservator based in the Houston area, and I know she's in the audience tonight.
Woo.
Hi, Kathleen. She has been instrumental in helping us understand what we're seeing, both under the microscope and when we get our analytical results back. It's so great to have a textile brain on the team. So she did a very close examination very early in the project, and we learned a lot of really key information. Michelle introduced you to camelid fibers, and I think those are some of the things that we expect to see in textiles from modern-day Peru. But what we were seeing under the microscope was not. It was absolutely cotton, and I'm going to show you how we know here. On the left is a polarized light microscope image. Essentially, we teased a few fibers off of the textile, placed them on a clear microscope slide, shined light from beneath it, and then we were able to kind of look at the morphology of the fibers and how the structure of the fibers interacted with light. A lot of the things you're seeing on this slide are very characteristic of cotton. So I hope you can tell it kind of looks like a flattened tube or a ribbon that's twisting over on itself. And when you see that, you know immediately this is cotton. On the right side of the slide is a scanning electron microscope image of a full yarn. And in this image, you can also see the twisting of the cotton fiber that is so characteristic, but you can also see how all of the fibers are brought together into a bundle and spun to become a single yarn. And some of the things that we look at in images like this are the diameter of the yarn, the direction of the twist, so like which way would that spindle have to have been spun when they were spinning the fiber, how tight is that twist. All of these things really have implications on how the fabric itself behaves, and also could give us clues about where the piece is from. So what does a cloth's construction tell us? We were introduced to the backstrap loom a little bit earlier, and we believe that the textile was made in 2 continuous strips, about 3 feet wide on this type of a loom. It would have then been stitched together along, I guess they're lined up on their long edges like this, and a whip stitch seam was made to create this kind of huge banner that could be used to create the prisoner textile. The weave structure itself can be described as a basket weave. It's a warp-faced basket weave, to be even more specific. Thank you, Kathleen. This is essentially a variation of a plain weave technique where you have 2 yarns worked together and they are woven over and under. So you go over 2, under 2, over 2, under 2. And this would have been chosen because it creates a really smooth, sturdy fabric with a heavy drape, something that would be an ideal substrate for a painted textile.
Like a canvas.
Like a canvas, absolutely. You got it. There's a detail in the center. I think this is one Fran and I took when we were looking at the DMA's fragment, and you can really see, Kathleen made a remark in her report about how consistent it was throughout. So the thickness of the yarns, the density of the weave, it was just all pretty standard from one end to the other, which is astonishing if you think about it. Taken together, all of these things are consistent with what we know about other textiles from the region, and that really helps us begin to confirm our provenance, what we know about its provenance. Before I leave and go to the next question, I wanted to leave you with a couple of these charming images. These are my favorite from the whole project, taken by Kathleen. In about 5 or 6 places, maybe even more, across the textile, she found these individual human hairs trapped in the weave. Can you guys see those? And especially the one on the left is really obvious. So you can just imagine these weavers bent over their loom and working really hard to get this like intense amount of fabric made. Their hair fell into the piece, was trapped, and hundreds of years later, we're looking at it under a microscope. And it's just, for me, it's an intensely personal thing, almost like a signature they didn't mean to leave. So I think they're wonderful. And thank you, Kathleen, for capturing those. How old is the textile? There are, you said 10 fragments, I believe, in collections across the world, and all of the collections give a slightly different date for their manufacture. And what we know of the dates is based on provenance information. But we wanted to take what we learned from the visual analysis I just talked about, which kind of gave us a general idea of the time and place, and drill down to a more specific date that we could, we could say, science told us this. So we were really happy to know that we could take a sample of the Menil's piece and get a date back, and that would, you could extrapolate that the date would be the same for all other 10 because they used to be a single artwork. So in science, when you have one result, that's great. But if you have a second result that repeats the same answer, that's even better. You can trust that even more. And here is, again, where the DMA came in clutch. They allowed us to sample their textile and send it in with ours. And what happens is you take just a few milligrams of yarn trimmed from, there's raveling edges where the Peruvian dealers in the 50s cut the piece apart. We just took a few of those and sent those in. How does one date a textile? Cotton is an organic material, and it can be, we can use a we can use a radiocarbon dating technique called accelerator mass spectrometry. And here's where I try to explain something that's more complicated than I am qualified to explain. But solar energy generates carbon-14, and it's incorporated into all living organisms when they eat and when they breathe. And at the moment an organism dies, that starts to decay at a known rate. So accelerator mass spectrometry measures how much carbon-14 is left in a sample today, and they can mathematically work out when the date for that material. It's usually given in a range of confidence, like we know 95% confidence that your date is between these two dates. We worked with Dr. Alexander Cherkinsky at the University of Georgia's Center for Applied Isotope Studies. So he actually did the analysis for us. On this timeline, you're looking at our understanding of the Prisoner Textiles date originally. So we thought this has been made between 12 and 1300. And again, that was based on provenance studies, and probably the iconography gave us some clues as well. That is overlaid on two blue bars that represent the general dates during which the Chimú and 𝗕𝗵𝗵𝗃 were flourishing in this region. When we got our results back, did you see that jump, that huge jump there? Hold on, let me do that again so you can see. Luckily, both the Menil's and the DMA's dates came back the same, so that's great, right? That's a win for us. But why am I so excited about this 200-year switch in the date? Well, what has happened is our new understanding of when the textile is made coincides perfectly with the Chimú conquest of 𝗕𝗵𝗵𝗃 in 1375. So this lends credence to art historical theories that the prisoner textile depicts these military victories. And it's also interesting to think about the fact that they co-opted the artistic styles of the peoples they were conquering to depict them, so we would know who they were. Here's an example of that. The center, the mask in the center is a funerary mask from the 𝗕𝗵𝗃, and the characteristic teardrop-shaped eyes are sketched out in the upper left so you can see them more clearly. This is shown alongside 3 different depictions of figures from the prisoner textile, and I hope you can see that they all have very similar shaped faces, eyes, noses, the way they've depicted the ears. Could we indeed be looking at a depiction of the end of the 𝗕𝗵𝗃 Empire? I think the dates that we got back from radiocarbon dating really do support that. So it's an open question, but we know a little bit more at this point. I'll hand that over.
Thank you. So then we went on to address other questions. Primary amongst them were, what were the colorants that the artisans used? And we've talked about taking samples for radiocarbon dating, and one of the ethics of the conservation and conservation science fields is we can take samples from areas of damage, but we can't take samples from areas that are not damaged in a way that harms the object. And all of our colored areas are on the interior of the textile, so we cannot sample from them, which means we have to turn to non-destructive and analytical techniques. And one of these that we use very often is X-ray fluorescence spectroscopy. Scientists love big, long names. And then we love to give them acronyms. So we're going to talk about this as XRF, because it makes us feel smart. So XRF is completely non-destructive. What happens is the instrument shines X-rays at an object, and the object absorbs some of that energy and may shine X-rays back. It fluoresces, just like fluorescent things fluoresce visible light. And different elements will fluoresce different X-rays. And by reading what X-rays are coming out of an object, we can tell something about the elements that are there. And being a scientist, I love pigments, and I love paints, and I love the bright colors, and all of the natural pigment colorants that the ancient Peruvians would have used, like red ochre, green ochre, azurite, vermilion. Those all have metallic elements. So iron in the ochre, mercury in vermilion, copper in azurite. Our textile has none of those. So none of the colorants are inorganic colorants. And so that means that instead we're dealing with paint that was made using organic colorants. And that makes our lives hard. Because it turns out that almost any living plant can be used as a dye. They may not all be pretty, but they can all be used as a dye. And so this is a photo taken from a modern Peruvian weaver's studio that shows different plants that are native to Peru and the yarn that can be dyed with those plants. So we needed to figure out what plants would have been available and what colorants and colors they might have produced. And for that, we needed references. And despite the Bank of America grant, they were not going to give us money to go to Peru and get samples of all the plants and all the yarns.
Boo.
Yes. It would have been really fun. But luckily for us, this amazing woman, 𝗕𝗚 did this back in the 1970s. She traveled through Peru in a Volkswagen Beetle, collected plants, noted where she collected the plants from, and then dyed yarn with those plants. And she assembled these all into this notebook that you see here. It's amazing. And this is just one page with all of these tipped-in yarns with all of the recipes she's used. It's phenomenal. And these notebooks live now at the University of California, Los Angeles in the Cotston Institute for Archaeology. And thanks to Dr. Stella Nair, who's the associate professor there, and Veronica Muros, who's the director of the Lab for Experimental Archaeology, we were allowed access to this notebook. We were also able to purchase this book, which had been printed in the 1980s, done by Hugo Zumboldt, Tintas Naturales para la Lana de Oveja, or Natural Dyes for Sheep's Wool, that has little tipped-in yarns that you can see at the end. And he's not as precise in his recipes as Kay was, but he does give some, and he does list the species of the plants, and they're accompanied by really charming little cartoons of how to do the dyeing. So if you ever get a chance to look at this, this is fun. Now, keeping in mind that we couldn't take samples, and we have to do everything non-destructively, one of the very few avenues of experimentation open for us was yet another fancy scientific thing, fiber optic reflectance spectroscopy, or FORS. Remember, we like acronyms. And basically what this technique does is it looks at what light wavelengths are being absorbed by an object. So we have a little piece of our Prisoner textile. We're going to shine white light on it. And the way that our eyes work means that whatever colors the object doesn't absorb get reflected back to our eyes, and we see it. So we see that hat as blue because it's absorbing every other light but blue. Now, FORS does slightly more than, hold on, wait, I'm going too far ahead. Here we go. Now, like I said, there's 9 million different kinds of plants that can make dyes, and it's particularly bad because of those 9 million, like 899,000 of them make red and yellow dyes. So this is just a small number of plants that we know create red and yellow dyes, and one insect in this case. And we cannot tell what the colorants are for the red and yellow on the Prisoner Textile. Yet. This is a continuing story. This is the never-ending story. I will spare you the song. But what we can tell is that the blue is indigo. So here we're looking at a graph which shows the absorbance when the line gets low and the reflectance when the line gets high of the blue in the Prisoner Textile versus a set of indigo dyed wool from the Zumble book. And they don't align exactly. One's cotton, one's wool, one's, from the 1980s, one's from the 1300s, but it's close enough that allows us to know that it's indigo, which makes perfect sense because indigo's endemic and natural, grows naturally in Peru. So no big whoop there.
So we could also see that indigo using infrared photography. As Cory just explained, the spectral characteristics of indigo in the infrared region can help us distinguish it from other visually similar blues that we might be encountering. So by taking a modified digital camera and capturing the textile with it, doing a lot of crazy post-processing, you can get what amounts to a map of where indigo occurs on the surface. So the image you're looking at now was taken by James Craven, who works at the Menil and is our amazing conservation imaging specialist. Sorry, James. Everything in his image that appears white maps to a location that is giving us the spectral response of indigo or something very similar. I think it's fair to say on this piece, we're not expecting to see anything different. So this is a map of indigo. And it corresponds really well to the places where we see blue figures, but also, because yellow and blue make green, where we're seeing green figures. What happens there is that the yellow figures were overdyed with indigo to create a new color. So you see that here on the map as well. And when you look even closer, you start to see that there's these like bright outlines. So indigo, it kind of traces around every one of the lines and figures in the textile. We'll come back to that in a minute. But let's talk about how the colorants were applied. We know from everything we've talked about so far that we're dealing with dyes and not mineral pigments that can be mixed into paints. So how were they able to get them on the textile with such control? When we think of dyes, we normally think of immersion dyes, so something that, a piece of fabric that you submerge in a boiling vat and it comes out a uniform color later. But given that we have these discrete areas of color on a blank, undyed background, and given the fact that they were able to mix them, that suggests to us that they were modifying the rheology of these paints, or how thick they are. To be able to get those hard edges, and I don't think I even explained this yet, but on the backs of these textiles are pretty much blank. So there's not a whole lot of bleed through of the dye to the back of the textile. And to do that, one would have to take their dye and make it behave like a paint. So how did they do this? Cory and I spent a lot of fun afternoons creating mock-ups using dyes that would have been available in the region at the time. And we were doing this not only to test different techniques, but also to be able to look at them with the 𝐹ORS instrument. We could take their measurements and add those knowns to the spectral library of Andean dye stuffs. So that's really great. We were testing, we made up a bunch of really concentrated dye mixtures, and then we were testing how we could thicken them and if that even worked. We did have success with gum tragacanth and guar gum, I believe, two naturally derived gums. And that being said, they were still incredibly difficult to control, so hats off to these artists. There are other ways that you can modify them, kind of like diluting those concentrated dyes or mixing them, and we tried those as well. One of the other avenues that we wanted to explore was whether there were any visual clues left on the surface that would help us determine whether something had been stamped or stenciled or hand-painted. So after all of these explorations, I think we're pretty certain that what we're looking at is indeed freehand painting. Not quite freehand, because remember those outlines I showed you that we saw in the IR maps? I believe that those were, let's call them cartoons, that were drawn on the blank textile to act as a guide for the painters. But after that, they had nothing else to do except for freehand paint these. We did want to be sure, however. We do love our confirmations. So we did task our imaging scientist. Why do I keep doing that? He is a scientist to me. An imaging specialist, James, he did some digital analysis of the piece. He was looking at the correspondence of all of the repeating figural elements and telling us essentially, were they regular enough to have been created by a stamp or a stencil? I think he came to the same conclusion that we did through our hours of just trying it. We're all pretty certain that this was hand-painted, so that's nice.
Can I jump in for a second?
Yeah.
So if you go upstairs to see the prisoner textiles on view after the lecture, take the opportunity to look very closely, because you can see the little idiosyncratic errors and oversights. There are outlined areas that are left blank. There are figures that are kind of crammed in, like, oops, maybe we didn't have enough room to do what we were supposed to be doing here, so we're gonna just make the best of it. So you can really see that human hand behind the textiles. And when you have something that is so monumental and yet you can drill down into a tiny little detail, like, oh, this double-headed snake was not painted in, it just makes you think about how intense this process must have been.
And then you also have to decide whether you're on Team Snake, Team Mouse, or Team Feline. So given that these are organic colorants, we then have to be really concerned about how light fast they are. We know, because we live in the bright Texas sun, that if you hang things outside, they fade. And the same thing can happen with any art material. And we need to know a little something about how delicate or how fugitive the prisoner textile colorants were. So the way to assess this is that first you have to have a standard. And it turns out that people have been doing this for a while, and they have developed what is called a blue wool standard. This is a card shown on the screen with 8 different blue dyes on it that have different levels of light fastness. 1 is the most fugitive, it fades the fastest. 8 is the most stable, it hardly fades at all. And so basically, anytime you want to know how light fast something is, you compare it to your blue wool standards, and then your curator yells at you because you told them you can't put it on display.
And we do yell.
So basically, you cover half the card. I mean, to give you an example of how this works, you would cover the card, you expose light, and then ta-da. The blue wool one basically goes away. So we don't want anything that's blue wool one. So we did this on a tiny, tiny scale. We did not expose the entire prisoner textile to light. Instead, we shone a very bright light that's a millimeter wide on one area of it and looked until we just began to see fading. And then we compared it to the blue wool card. And what we found was that the most, here's the little tiny light, the most fugitive color on the prisoner textile was about equivalent to a blue wool 3, which is not great. It does not make us happy. We cannot put our fragments on long-term display. It's why when you have been up or when you go to see the room where this display is, you'll notice that the lights are low. We're protecting these. But also, these fragments have not been on view very much. And so we felt comfortable saying, yes, it's important for people to see them, both at the Menil in the Enchanted exhibition, and then for lending to the DMA, because it was so important to get these 2 fragments back together.
And I think this is the first time since 1951 that two fragments of the Prisoner Textile have been reunited, if you will. Thank you, Fran. They're not like placed next to each other because they're not in the sequence next to each other. But it's pretty remarkable that we have two of the fragments here in Texas. You know, a big dream would be to have all 10 fragments reunited, but that would probably take like 10 years of our lives to execute, and
End of the project.
yeah,
We'll do it for the end of the project.
yeah, there we go.
So in addition to all this research, we also had to do the practical thing of preparing these textiles for display. They have since been on 2 exhibitions, and we've thought really hard about how to do that safely. But an important part of the grant project was the conservation treatment of the Menil textile fragment. And this was done by Kathleen Kiefer. You can see her here working on one of the aspects of her treatment. And now, I'm not going to remember all of the different things that she did without written notes, but she cleaned the surface. I know that there were lots of creases from past storage folded that needed to be humidified out. All of this she's doing while being very careful about the water-sensitive pigments. She did not want to create any more bleeds on this piece, so great finesse. There was several sections of the central, remember the horizontal seam that joined the 2 3-foot fragments together? So parts of that needed reinforcement, and she did that. And there was also a beautiful stitch that she put down each edge. The edges, remember, that were cut by the dealer, so they would not ravel any further. You can also see her here making a custom tube. It's an archival tube, and it's padded. It's a brilliant, beautiful thing, and it's a very important part of its preservation strategy, although it's something that all of us don't really think about. So thank you to Kathleen for all those efforts. Yeah.
It's like sleeping on a Tempur-Pedic.
Kind of, yeah. It protects the textile from dust and light when it does not need to be on view and exposed to those risks. So she and I also worked with our very talented matter and framer at the Menil Collection, Mina Gaber, to design and fabricate an enormous frame for this thing that has UV filtering acrylic on the front. And that protects the piece and allows visitors to approach it very closely and really get down and see those details that we've been talking about. It protects it from dust, and it really does shield those fugitive pigments from light. It's now been in 2 exhibitions in this frame and has done beautifully. So on the inside of the frame, what you're seeing is the backing board is made from a plank of archival foam that is covered with a heavily napped fabric. Think of like a suede or something like that. So it's really grippy. We were able to pin the textile to this using insect pins, and these are things that we borrow from entomology, but they're just, think of them as very thin stainless steel pins. They were inserted between the threads every few inches across this entire textile. So there's dozens and dozens of them. And together with this grippy backing fabric, we've essentially given the piece an incredible amount of gentle support that's invisible. You'll see, you can go back upstairs and try to find some of the pins. They're very carefully hidden in design elements where it makes it more difficult to see them. But yeah.
And when we were mounting the exhibition, it took you 2 hours to put all the pins in? And
yeah, about half a day.
half a day total. Mm-hmm. And then our section of the textile is half the size, so it really took us only about 2 hours.
Yeah.
Yeah.
And we're just grateful that we have a way to present it vertically, which is, the way it probably would have originally been shown, without putting any additional stress or causing tears or sagging.
I think this is where we pop to the DMA lifting their idea and implementing it here in a less, can I say, bougie way, because we didn't have a Bank of America grant, but we know we're scrappy and we know how to get things done here. So we replicated their technique with other materials, and every single textile that you see in the exhibition upstairs is hung using this pin mounting technique, except for the one tunic in the show, which is hanging on a dowel. And Mary Nicolette and Fran Boss were able to pin the textiles pretty rapidly. It was pretty amazing to see how quickly they were pinned onto these backing boards, and they just look so beautiful. It's such an innovative and lovely way to show these fragmented works, and again, to echo what you said, give them the support that they need while they're hanging vertically. And hats off to Mary, who painted, I don't know how many hundreds of insect pins, which you can see on the boards here. And then you can see in the upper right-hand corner kind of all of the pins sticking out of one of the textiles before we popped them all down. But it's a really awesome way to hang ancient textiles, and the backing boards that we created, we hope to recycle here in future exhibitions across the museum, different collecting areas. It's almost like the clouds parted and the sun shone down, because now we have this awesome way to hang these textiles. So thank you, Kathleen.
So one of the questions that's still very much ongoing is, what is the staining? And Andrew Hamilton at the Art Institute of Chicago had proposed that it might have arisen because the textile was placed under a mummy bundle, and that as the body decayed over time, it leaked mummy juice onto the object, which then hastened the decay of particularly the AMNH, American Museum of Natural History fragment, and stained through. Now, this is where we had to go to a real scientist. So Caroline Tokarski at the University of Bordeaux is one of the premier scientists when it comes to protein identification. And we were really fortunate that because of our past association with Julie Arslanoglou at the Metropolitan Museum of Art, we were able to convince Caroline that this would be a really great project to be involved in. She fell for it. So again, I'm going to get a little bit technical here. So amino acids are the building blocks of life, and they are used to create proteins, which are what run the world, or at least run us. Proteins can be very big in a molecular sense, not in an actual, inches or centimeters sense. And so when scientists go to analyze the sequence of the protein, how the amino acids are arranged, they usually have to cut a protein into little smaller bits. And those smaller bits are called peptides. So I'm going to talk about peptides and proteins. So what Caroline does is she takes samples that we sent her, and I'll get to that in a minute. She and her lab extract the proteins out. They cut the proteins into peptides. They separate the peptides, and then for each peptide, it gets chunked into a $2 million piece of equipment, not jealous, that tells her the mass of each peptide. Now, the mass allows her, with the help of lots of computers, to know what amino acids are present in that peptide, which is interesting. But it's not enough to tell us what sequence those amino acids are in, the exact order. So instead, this machine takes that little tiny peptide bit that it knows the mass of, and it breaks it down again into smaller fragments. And again, each of those fragments we know the weight of. Therefore, we know the amino acids, and by doing a lot of computer reconstructing, we can then say that this peptide fragment weighs this much, this peptide fragment weighs this much, this peptide fragment weighs this much, this has to have this amino acids, this has to have this has to have this, and then reorganize that and figure out logically what the peptide sequence was. This is like the biggest game of logic in your life. If you love Sudoku, go become a proteomics specialist. I am not a proteomics specialist. But this is done for each peptide in a protein. Then it's done for the next protein, and the next protein, and the next protein. So running these analyses is hugely complicated. And so that's why we don't have a lot of information from Caroline yet to share with you, because she's still very much involved in data analysis. The other issue we had, again, was sampling. So again, we can't take chunks of sample from the middle of a textile. And our fragment at the Menil is one of the ones from the end of the textile that doesn't have any staining. So once again, we turned with love and hopefulness to our colleagues here and asked if we could work with them to sample their textile. But we had to do it non-destructively. And that's where we stole a page from people who look at parchment. So people who look at parchment books have been very interested in figuring out whether the parchment comes from sheep or from cow and blah, 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 blah. We're not interested in parchment, or at least not in this project. But the way that they tell that is that they use eraser crumbs and they rub them on the parchment, and then they analyze the material that's come off on the eraser crumbs. Now, paper conservators do this all the time to clean paper objects. It's considered okay. They would normally just throw the eraser crumbs away. But for anything that has proteins in it, those eraser crumbs become very valuable sources of protein. And Kari and I did mock-ups, which are disgusting, and I will not tell you about them, and sent samples of eraser crumbs rubbed on canvas to Caroline, who was able to show that, yes, we were with eraser crumbs picking up protein from the surface of cloth, largely non-destructively. And so Kari came and was able to, with Fran and Michelle, which is the photo that you saw at the very beginning.
Hive mind.
Yes, hive mind photo. To sample using eraser crumbs from the back of the DMA's fragment. So those are what got sent to Caroline. We've also, and here they are doing it again in our little like Eppendorf tubes full of eraser crumbs, and it's so fun. It's very satisfying. We were also recently given permission by the American Museum of Natural History to take samples from their fragment, which is the most stained and the most degraded. And so Caroline now has fragments, larger, like actual fibers from this, that she can compare to the eraser crumbs. And like I said, this is very early stages of data analysis, but she's already very easily been able to show that the cotton is a Gossypium hirsutum, which is endemic, native to all of the Americas and expected, and that she's got a ton of proteins to look at, proteins that come from humans, proteins that come from bacteria, proteins that come from fungi. All of these things that she's now trying to sort out what species they are and what they can tell us about the environment of the textile, what caused the staining, where it's been in its life, keeping in mind also that we've all been handling these, and the looters handled it, and the people who wove it handled it, so we're getting information from all kinds of humans as well. So it's going to be a long, complicated thing, but maybe in 10 years we'll figure it out.
Come back for part 2.
That's right, and we'll get more of the fragments together.
And we're done.
With immense thanks to everybody on this slide and to all of you for your attention.
Yes, and we've tried to thank people as we go along, but there have been, as you can tell, a lot of people involved in this project, and it's been a real privilege to do that and to become really close colleagues and friends with our partners here.
Yes. Amazing. I think right now we have the opportunity to take some questions from the audience. Michael has a microphone that he is happy to rush to you if you have a question.
Hello, I'm Romina Levy, and thank you so very much, because when I passed through that section of the museum, I appreciate it, but I'm not really, I don't really know how to, I don't know how to admire, besides admiring it, I don't really know what I'm looking at, so thank you very much. I sound rather ignorant, but I have this question for you, and it's really very basic. So you said a number of things. You said it's, in its entirety, it's 75 feet long. At one point, you also mentioned that the staining is probably because there was seepage from burial or
A mummy bundle.
a mummy. So here's this altogether, not very articulate, but was it therefore possibly done to depict the political climate, maybe, of the time? Was it advocating in favor of what was happening at the time, or as a resistance against what was happening at the time? Was it done for burial purposes? I guess that part threw me off. I sort of, not you lost me, it lost me at some point.
That's fair. That's fair.
Thank you.
This is a very good question. Thank you for asking it. Because we don't have a controlled scientific archaeological excavation in which the prisoner textile was recovered, we can't maybe know as much as we otherwise could. This is why these kinds of technical analyses are really important for museum collection objects that don't always have that archaeological provenience or find spot that we can gather information from. And you know, we might look at the textile and think, okay, the amount of labor and effort that went into producing it suggests that whomever it was buried with was extremely important. You know, a lot of resources were expended to create this textile. And I think Andrew Hamilton, who you mentioned earlier at the Art Institute of Chicago, I think he believes that it was specifically created for burial. This is up for discussion. We may never know. We also may never know more about why it was created than we currently do. So if it seemed a little fuzzy and vague, it's not because you misunderstood, or it's because it is fuzzy and vague. And that's kind of the reality of the situation. So thank you for allowing us to kind of dwell on that a little bit more.
Have you found textiles like this in an archaeological context before?
So I recently, and you may also want to jump in depending on your conversations with research specialists, but I was recently in email exchanges with somebody who is presently excavating in this region and is uncovering painted textiles in the excavations. Historically, there haven't been a ton of excavations where painted textiles have been revealed. So we don't have a lot of archaeological comparatives at this moment, but you know, ongoing research is providing us with those.
From the analysis and spectrometry of the cotton and colorants, specifically the cotton, was there any evidence that maybe it was from a time of drought or a wet season? Is there any comparable samples you could have placed to see if that was an evidence?
I'm not sure that I know a way to tell. I'm sure there is someone out there that we can ask this question of. I'm thinking a little bit about one of the other observations that Kathleen made during her analysis of the piece at the beginning of the project, she noted that the lumens, or the channel inside these cotton fibers, seemed to have some sort of dark colored material in them. And there's still, it was just an interesting observation. I don't know what it means. It could be indicative of the species of cotton that we're looking at, which then could give us information about what time of year it might have been grown or what conditions were around. But it's just another question to ask, and we would love to do that at some point.
Unfortunately, cotton, with trees are nice, right? They grow or they don't grow, and they've got their life history in their trunks. Cotton is much more short-lived, and it tends not to record a lot of that in the same way, which is unfortunate for us, because that would be great, right? Sometimes times of warfare happen during times of climatic upset.
This is a little bit of a historical context question, but I want to know if we have any information about what these people may have called these colors as they were painting them, and if they referenced, whether if it was blue, green, or yellow, or if it referenced the materials that were used to paint these colors.
I like how you look at me.
Well, I don't speak Quechua,
I
which
don't
is the
speak
closest
Quechua
living
either.
analog.
So Quechua is the language that's presently spoken in that area, and I don't know that that's a question we can answer, especially without a written record of any kind that allows us to compare past to present references. So I'm going to draw on my own expertise in the Maya region, particularly in Guatemala. The Maya were the only culture in the ancient Americas to develop a written script. And that gives us a little bit more of the kind of insight that you're looking for, but it's still like a huge guessing game. So I think the only genuine way to know would be to time travel, which would be amazing, but not happening yet.
And not exactly what you're looking for, but a lot of the references we mentioned that we were finding that had examples of dyed cottons tipped into them did give us names that were used by the people in that area at that time. Granted, this is hundreds of years down the road from when they were made, but I thought it was fun to see those and to know, what they were called to the people that use them.
Yeah. Kay recorded the Quechua names for the plants when they were specific. And actually so does 𝗏𝗏𝗏. So we've got a lot of the more, at least modern plant names that refer to them, but.
Hi.
Hi, Tara.
Hi, Michelle. I have a mummy bundle question for you. Shocking. Okay. You mentioned that this is the painted fabric. What about comparisons, and I'm probably thinking different cultures perhaps, of woven fabrics and the length of woven fabrics around mummy bundles, which I know if they're found, they can't always take off because of they disintegrate. But do you know about woven fabric lengths for mummy bundles?
So I think that's an interesting question, but I think there's probably a little clarification that I can offer. The mummy bundle itself would have been wrapped in its own material. And so this as you can see kind of in this drawing, there's an even more plain outer wrapping that Andrew portrays in his watercolor of the mummy bundle. And I think that the Prisoner Textile was kind of an ancillary cushion. It wasn't necessarily created to shroud the mummy per se, but was used around an already wrapped mummy bundle. So I think that's a really complicated question to answer, but this particular textile itself wasn't used directly to wrap a mummy.
Hey there. You mentioned the surviving Andean tradition of weaving, and I'm curious if there's surviving traditions of textile painting in the region as well, and what you know of that.
I'm not a Peruvian expert, but the little bit I know, it's not been kept alive the same way that the weaving tradition has been.
That's my sense of it too. Although, you know, there could be artists somewhere who are definitely still producing work like that. But by and large, the textiles that you see on the market, you know, created by contemporary artists are dyed woven textiles, not painted.
Hi, thank you. My question's about the hair samples that you guys encountered in that fabric. So obviously it's not relevant to your project, but it seems like there might be some value in sequencing that in other scientific kind of endeavors, looking at those cultures and that time. Is that something that's been considered, or has it been published internally that those are there? Just because that seems like a really interesting finding.
Yeah, their presence has been duly recorded. Thank you, Kathleen. And like I said, this project has started with a bunch of questions and begat even more questions. And this is certainly one of them that could be explored. For now, those hairs are safely tucked away in the textile. Maybe you can go upstairs and try to find them, connect with them on a personal level like I did. But
yeah, we'd have to, protein hair is keratin, and keratin is kind of not super characteristic other than human. And so we'd have to think about DNA sequencing of them, and that's becoming a lot more feasible these days with improvements in sensitivity, but we'd still have to have something to compare them to, and then you get into health and human privacy issues, and we'd have to think about whether the people still alive there had any interest in knowing if there was a linkage there. And so it would be an interesting project and might help people reconnect to this textile, but it's pretty different.
Unfortunately, we have time for one last question. Hopefully our speakers will stick around for a little while. You can speak with them, but here we go.
Between the separating the pieces and the degradation, are we confident that this is the entirety of the piece?
I think there's been several thoughts about this throughout its the history in the literature. So people have been, there's a slide that Michelle presented early on that shows the order of them. This is not what we always thought. Andrew Hamilton, who we keep bringing up, working at the Art Institute of Chicago, did a great paper in 2020 where he proposed a new resequencing of them. And during that time, I think the previous thought was that we were missing several borders and additional panels, and he kind of took a look at the staining and the ordination of the color, you know, the way the colors were patterned. You know, there was a determined order that the colors went in. So he looked at all of this and put together a new thought about how long the piece was, and that only includes maybe one missing panel and one border.
There's an alternate reconstruction that he's proposed where there's a thin missing panel just of figures that would have edged one of the scenes, and that kind of changes the whole like last half of the textile. But you can see when you look at this, it kind of feels like a Rorschach image where the staining is pretty regular and clear, and so that was one of the huge clues that he used. Every other frame, if you will, has the mouse.
And you can tell that the Princeton piece is missing its left border. So that's one piece that could potentially be missing. Everything else has that mouse. Is it a mouse? A mouse border? Yeah.
I also want to point out, back to Dapper John Wise, there's a textile next to him that is also painted, but this one is called the Marine Fauna textile. And originally, the distinction between the two was unclear. There are no pictures or records of what the textiles looked like when they were discovered. And so the only thing we have to go on is how they look now and how we might hypothetically reconstruct them. The marine fauna textile, I don't know much about it at all. It's kind of elusive, and there aren't very many fragments of it left, but everyone thought that it was a single big textile initially. So our understanding of the textile has changed over time. But hopefully, you know, as time continues to pass, we'll understand even more.
Well, please join me in giving a warm thanks to our distinguished speakers.
And I want to thank everyone again for coming out on this Saturday afternoon and for joining us at the 𝗏𝗏𝗀𝗃 Archaeology Lecture Series. And we're looking forward to booting up another archaeology lecture in the fall. So come back.